Good morning. Welcome back to Biology 1301, the biology course for non-majors. Today we're going to talk about Chapter 6, DNA, Structure and Function. Everybody's heard the word clone. We've all talked about cloning. You've heard about cloning. You've seen science fiction movies that deal with cloning. What exactly is cloning? Cloning is the technology that produces an exact genetic copy of an individual, and that is referred to as a clone. In a very sort of broad sense, identical twins are clones of each other. They start out as one fertilized egg. That egg separates, and then two individuals grow from that separated egg, that separated fertilized egg. And they are genetically identical, ergo, they are technically clones. But in most times when we think about cloning, we think about taking an existing organism and making an exact genetic copy of it at some point after birth. And again, those science fiction movies are always talking about some rich guy wanting to clone himself so he can basically live forever. And I'm sure there was a James Bond villain at some point who was a clone or tried to clone somebody. But to make a clone from an adult cell, you must reprogram the DNA in that existing cell to function like the DNA of an egg. So you have to take cellular DNA and make it act like the DNA would act in an egg cell. Somatic nuclear transfer refers to taking the DNA from some adult somatic cell. And by somatic cell, what we mean is any cell in the body other than the sex cells, the sperm and ova, or the egg. That somatic DNA is then transferred into an unfertilized, unnucleated egg. Unnucleated means through scientific processes, we have taken an egg and we have removed the nucleus from that egg. Then we put that nucleated egg in we take the somatic cell transfer DNA, place it into the egg, and then stimulate that egg to start dividing. This is now somewhat common in livestock breeding. The people who deal with dairy cows and beef cattle, they have, once this technology has been gotten to the point where it's viable, they have found that it's easier to take a cow, a really well-producing milk cow, and making a genetic copy of that rather than breeding that cow and hoping that those genes that made that cow be so much better at producing milk are passed on to the new offspring. You can take a female cow and you clone it. You're going to have a female cow. Well, if you take that same female cow and breed her, there's a possibility you're going to get a bull rather than a cow, and you can't get any milk from a bull. So that's the reason this is becoming more and more common every day in livestock. Cloning mammals is unpredictable. The way they do this is they don't just implant one egg, they implant a bunch of eggs in the hope that one of those eggs will attach itself to the wall of the placenta and develop to a new offspring. But very few of those implanted embryos result in live birth. Many of the clones will have very serious health problems. That's one of the drawbacks to it. As our technology gets better, I'm sure the number of serious health problems will go down. One of the first mammals to be cloned was a sheep by the name of Dolly. You can just go into your Google and type in Dolly the sheep, and you'll see all sorts of stuff about Dolly the sheep. And one of the things about Dolly the sheep was she only lived about half as long as a normal life expectancy for that type of sheep. And most of the researchers feel it was because there was some problems with that transferred, DNA. Here is what happens in somatic cell transfer. If you look at this top picture, there is a little blue sort of structure right here. That is the nucleus. That's what they're going to remove. This is a very fine micropipette. This is very, very tedious, very technical techniques. You're not going to do this in your garage at home. It takes, it takes special training to be able to do this. But what this technician is going to do is he's going to insert the needle. Then they're going to remove the nucleus. And you can see here, I would have much preferred if they had taken their picture with the nucleus right about here where they were removing it. But you see this little blue dot that is the nucleus is no longer in this cell. And then in the third view here, here's the nucleus of the cell that they've taken from. This is the transfer DNA, and they're going to inject that back into that cell and then stimulate it in the hopes that they can produce an offspring. Again, here's the continuation. We injected the new DNA. There's the new DNA. We stimulated it, and now we started to produce a new offspring, and you can see we're starting to get separation there into what will eventually be the new offspring. Just some examples of some things that have been, and take mine. This book is a 2016 version, so this is all 5-year-old data, so we'll keep that in mind. But everybody remembers the tragedies of 2001, September 2001. There was a dog. The dog's name was Tracker. He was a very good dog at finding victims. So good was he that it was decided by his owners and handlers that this dog should be cloned. Here is the clones. Here's the 2, or actually 3 little clone puppies. It's hard to see on this view, but there's a puppy right there, there's a puppy right there, and there's another one there. There's Tracker. Those 3 dogs hopefully grew up to be as good a finder of lost people as was Tracker. Champion dairy cow Liz on the right, and her clone Liz 2, also produced by this same method. Liz 2 had already begun to win championships in milking by the time she was a year old. I'm not sure what goes on in milking championships, but I would assume, how much milk did she produce in, a given time frame. They monitored the cows, they milked them, and then 12 hours later milked them again to see which one has the most. If you're really interested, I'm sure you can probably Google Liz and milk championships, and they will tell you more details about what goes on in a milking championship. Some of the history of DNA. There's a couple of names that are going to come up in a minute, and I'm going to point these out to you when I get to them. But the first person to describe DNA was a gentleman by the name of Johann Meichner. He didn't know what the function was. He just saw that there was this sort of an unusual structure within the nucleus of some cells, and he wondered what that is, and he described it, said what it kind of looked like, but he didn't know what the function was. Frederick Griffith discovered that bacteria contained a transforming principle. That transforming principle is called a plasmid, and in a later chapter we'll talk more about plasmids and how they work. And he discovered that there were capabilities where harmless bacteria could be changed into lethal ones, but he didn't really know, again, didn't know what the factor was going on there. He just knew that there were bacteria that under normal circumstances, these bacteria were completely harmless. But sometimes those bacteria would cause illness that literally could lead to death. The Avery and McCarthy, the 2 researchers who showed that DNA was the primary transforming principle within the bacteria. Again, there's also that thing called a plasmid. We'll talk about that a little more later. But Avery and McCarthy said that this molecule, whatever it was that Meichner discovered, and then that Frederick Griffith made a little bit more study of, these 2 researchers discovered that it was actually something called DNA. DNA must be able to do things to be considered the inheritable material. So in anything that is inherited, anything you got from your parents has to meet all of these criteria. First of all, it must be transmitted in a full set. The whole entire set of information needs to be transformed or transmitted. Now, in bacteria, that whole entire set comes from the bacteria. In you and I, we get an entire set, but we don't get the whole set from one parent. We get half of it from mom and half of it from dad. We get the whole set, but it's coming from two different places. So depending on the type of organism you are, you may be getting half from mom, half from dad, or half from one plant, half from another plant, but you're getting the entire set. It must be found in the same amount in cells of a given species. All of your cells have the same amount of DNA in it, with a couple of exceptions, and we'll talk about those exceptions. Remember, day one, I said, when you use the word all, always, and every in a science class, there's always going to be an exception, or there's almost always going to be an exception. So, virtually all of our cells have the, excuse me again, where is that there? All of our cells virtually have the same amount of DNA. This DNA must be exempt from major change. Now, it changes slightly from generation to generation, to person. My children have, they don't have the exact copy of my DNA. Some of them might, but some of them, and I'm saying that, I don't have any natural children of my own. I've raised 3 nieces. Well, I've raised 2 nieces, and now I'm in the process of raising another one that we've adopted the last one. But her father gave her DNA. Now, it may not necessarily be exactly the same as his. Could be, but there might be some slight variations. The difference is it must be exempt from major changes. It must be capable of encoding a huge amount of information. How much information? Enough to build an entirely new individual. You have to have, in every cell in your body has the entire blueprint to make another human being. And to make another human being just like you. The thing is, most of those cells, when they reproduce to produce more cells, when your skin cells, when they start making more skin cells, they only use a very small portion of that DNA. They don't use the whole thing. That's the reason this somatic transfer works, because the entire blueprint is there. Hershey and Chase confirmed DNA meets the first criteria in 1940. They did this by using what are called bacteriophage experiments. A bacteriophage is a virus that only infects bacteria, ergo the name bacteriophage. And what they did was they took these bacteriophages and they radio-labeled the DNA, and they then took radiographic photos of the DNA as it was moving from old cell to new cell, watching what went where, and they were able to figure this out. Here is a drawing of how that worked. The little thing that looks kind of like a spaceship is the bacteriophage. You see it's been coated with a specific isotope of sulfur. The DNA was injected into the bacterium. Excuse me. The bacteriophage exits. I'm gonna get it right in a minute. The bacteria has the new viral DNA within it. Here is a virus labeled with a phosphorus isotope. And you see here they labeled the DNA. Here they labeled the protein code. What they wanted to know is, where's the information changing? What's going into the new cell? What's not going into the new cell? And as you can see here, the bacteria, or the bacteriophage is here. Here's the new DNA that's been inserted with the label. And then this again exits, leaving the new DNA. So here you see on this one, the labeled product stays outside the cell. Here it moves into the cell. And this is an actual photo micrograph of bacteriophages doing their work. This is the detailed labeling done by, again, Alfred Hershey. I have no idea whether he's a member of the Hershey chocolate family. I don't know. Martha Chase, nice name to remember. Martha Chase. Keep your eyes out for that name later. This where they published their experiments with the bacteriophage, showing where the bacteria and where the DNA was going. Edward Chargaff discovered the relationships between DNA and the bases, and we'll talk about those nitrogenous bases in just a moment. Rosalind Franklin discovered the structure of DNA by using X-ray technology or X-ray crystallography. Again, another name to keep in mind for just another couple of minutes, Rosalind Franklin. Maurice Wilkins did experiments to show DNA structure. And then James Watson and Francis Crick built the first accurate model of a DNA model. Whenever we talk about, and whenever you Google who discovered DNA and kind of quote the word discover, you always see the names James Watson and Francis Crick. They actually won the Nobel Prize in, I think, biology, but in one of the sciences, maybe chemistry, in the 1950s for their discovery. A DNA molecule consists of two strands of nucleotide monomers running opposite directions and coiled into a double helix. If you think about taking a ladder, a long straight ladder, and twisting it, that's what the double helix looks like. If you take the DNA double helix and take the twists out of it, it would just look like a really, really long ladder. A DNA nucleotide, the monomers that make up the polymer that is DNA are referred to as nucleotides. One 5-carbon sugar, in the case of DNA, that 5-carbon sugar is deoxyribose. There are 3 phosphate groups attached, and there's 1 nitrogen-containing base, and there are 4 of them, and they are adenine, thymine, guanine, and cytosine. This is adenine. Here's the sugar. Here's the 3 phosphates, and here's then the nitrogenous base. All these nitrogens is the reason it is referred to as a nitrogenous base. Pay special attention to this structure right here. Notate how many ring structures there are. In adenine, there are 2 ring structures. Next structure is thymine. Again, 3 phosphates. Here's your ribose sugar. But now, for the nitrogenous base, you only see 1 of these ring structures. And this one is thymine. Then we have cytosine. Again, 3 phosphates, the sugar, and the nitrogenous base. Take note, only 1 of those ring structures. And lastly, we have guanine. 3 phosphates, sugar, and now we're back to 2 of the ring structures. If you recall, we talked about 2 different sugars that had 2 ring structures, adenine and guanine, and then we talked about thymine and cytosine having 1 ring structure. Now, Chargaff's rule, researcher by the name of Chargaff looked into this 4 different nitrogenous bases, 2 with 2 ring structures and 2 with 1 ring structure, and he said, how are those things going to orient themselves so the sides of my ladder are straight? And what he discovered was that the bases of the 2 DNA strands in a double helix pair in a very consistent way. Adenine always pairs with thymine. Cytosine always pairs with guanine. The proportions of A's and G's vary among species. Humans may have more A's than tigers. But no matter which ratio you have, A always pairs with T, C always pairs with D. And if you click back through your slides, you will see that that is 1 2-ring structure, always complementing a 1 ring structure. You've always got a 2 with a 1. 2 double helix strands are held together by hydrogen bonds between nucleotide bases. So the bond holding this two different sides of the DNA molecule together are hydrogen bonds. What do we remember about hydrogen bonds? They always come in large numbers. Well, if you've got a DNA strand that contains millions of bases, then you're going to have millions of bonds. And what is the other thing we remember about hydrogen bonds? They're very easily broken. So when the DNA needs to separate to form the templates to make new DNA or to make RNA and proteins, those bonds are broken very easily, but then they also reform very easily. Franklin and Wilkins separately worked on identical samples of DNA. Wilkins believed Franklin was his technician and not properly differential as one. Franklin believed Wilkins was prickly and dismissive of her work. So again, one of those names that I told you to remember was Rosalind Franklin. Where was she? Rosalind Franklin and Maurice Wilkins. So these are the two folks. Now, I wanted you to remember, Franklin provided critical data about DNA structure. It was her X-ray crystallography that showed all of these details. It showed the phosphate backbones on the outside of the double helix. It showed how the bases arranged in unknown ways on the inside. They weren't sure what was holding the bases together. Crick, of Watson and Crick fame, understood the data better. He realized that the nitrogen-containing bases paired on the inside of the helices. That's where he figured out that. And he figured out that they paired according to Chargaff's rule. So Rosalind Franklin did a ton of work into the structure of DNA. She didn't fully understand what was going on, but she did the work. She laid the pavement. She put everything down. Crick came along and was able to figure out the details. There's Watson and Crick on there. I believe that says that's a 1968 picture, but that is their DNA model showing all the different, you look at these little ball models that you do in chemistry. Each one of the little colored balls represents a different element. And you get all the hydrogen bonds here forming, and then the phosphate group on the outside. Now, Watson and Crick proposed that DNA consisted of two strands of nucleotides running in opposite directions, and they were coiled into this double helix pattern. They proposed that it was hydrogen bonds between the bases holding the two strands together, and that only two types of base pairings form, A to T and G to C, which ultimately became known as Chargaff's rule. The order of the nucleotide bases in DNA is the genetic information. A, T, C, G, G, C, A, T. All of those combinations make different proteins. And the two strands of DNA are always complementary of each other. And this is a strand of DNA that's had the twist taken out of it and had all the wonderful, shows the base pairings. You see here, T is always pairing with A, G with C, here's C with G, there's A to T, so you always see that same pairing. DNA in a simple human cell and a single human cell would be about 2 meters or 6.5 feet long if it was stretched out. Chromosomes are the structure that carries the DNA within the cell. The chromosomes can be as a condensed structure of DNA inside the cell. We only really refer to it as chromosomes when the cell is attempting to divide. At other times, it's known as chromatids and some other things. It's twisted in coil form, offers protection as well as saves space. And a histone is a spool of protein that the DNA strand wraps around inside the chromosome. Again, this is a structure. This is an actual photomicrograph of a chromosome in its condensed form. At its most condensed, a duplicated chromosome has an X shape. This will be, say for instance, this is chromosome number 10. This would be the copy of chromosome number 10 you got from your mom, for instance, and this would be the copy of chromosome number 10 you got from your dad. And the reason they don't show to be mirror images of each other, you'll notice this one looks a little bit longer. It's only because this one is just coiled up a little tighter. You know, everybody at some point in their life has had to, you know, loop up a garden hose, and you know, one time you roll it up and maybe it takes 10 loops, and the next time it takes 12 loops. And so that's what's going on here. This one's just twisted together a little tighter than this one. Over here, the artist rendering number 1 shows the 2 strands of DNA in their double helix form. At regular intervals, the DNA will start to condense. It will wrap around the core of histone, which is the purple structure. You see, that's the histone there. That's what starts to pull the DNA all together. Number 3, the DNA and the proteins associated with it twist tightly into a fiber. The fiber coils and then coils again to form a hollow cylinder. At its most condensed, where it's all twisted up together, you see on the right, and it's drawn here. And the DNA in the nucleus of a typical eukaryotic cell is divided into a number of different chromosomes. And we will talk about what goes on in the cell as the DNA is replicating in a later chapter. Duplicated chromosomes. When a cell prepares to divide, it duplicates its own chromosomes. Once that has happened, you're going to have 2 copies of chromosome 1 you got from your mom, and 2 copies of chromosome number 1 you got from your dad. You'll have 2 copies of number 2 from mom, 2 copies of number 2 from dad, et cetera, et cetera. Those 2 pairs, that pair of chromatids that you got from mom that duplicated, those 2 are referred to as sister chromatids. One of each attached members of a duplicated eukaryotic chromosome. Along the structure, if you look back at this, the structure right here where the 2 different chromosomes of the pair are sort of stuck together, that is a structure called a centromere. It is a constricted region in the eukaryotic chromosome where the chromatids are attached. You know, later on you'll see some of these that have very short top tails and very long bottom tails. Some of them will look, you know, like they're almost X's. So again, that centromere will form, and that's what causes that little constricted area in the middle. Again, this is a duplicated chromosome, just an artist's rendering showing you the unduplicated chromosome. In the duplicated chromosome showing the centromere here in the middle, you've got 1 chromatid and its sister chromatid. Again, actual photomicrograph of some DNA. This is chromosome in its condensed form. This is a chromosome in its uncondensed form. Chromosome number. All organisms have a very specific chromosome number. If you were to ever take a genetics class, for instance, and in genetics lab, one of the things we've done for years is we manipulate a little fly called a fruit fly. And the reason we use the fruit fly is because it has 4 chromosomes. So its chromosome number is 4. So it's a whole lot easier to work with something that has 4 chromosomes than human beings who have 46. Now, this is another number that will give us some problems because some authors will talk about human beings as having 23 pairs of chromosomes, where other authors will say human beings have 46 chromosomes. Well, 23 pairs is 46. It's just, you know, difference in semantics with particular authors. Chromosome numbers is the sum of all the chromosomes in a cell of a given type. The human body has 46 chromosomes. And when a cell has a complete set of chromosomes, we refer to that as being a diploid cell. So cells having 2 of each type of chromosomes are denoted by the characteristic number 2n. N being the number of chromosome types. And that's where that 46 versus 23 comes in. The human being final number is 46, So the N number for human beings is 23. We have 23 different types of chromosomes. But since we have 23 pairs, we have 46 total. Autosomes, all the cells in your body that are responsible for everything other than reproduction are called autosomes. These are the paired chromosomes. They have the same length, they have the same shape, they have the same centromere location, and they contain the same genes. Now, just because they have the same genes doesn't mean they carry the same information, and we'll talk more about that in a moment or in a subsequent chapter. The gene is the same, but the trait may be different. You'll have at the same point on the same chromosome, you're going to have a gene for eye color. It's going to be the same in everybody, but some people may have a gene for blue eyes, and some people have a gene for brown eyes, and some people have a gene for green eyes. Information may be, the specific information may be different, but the gene itself is the same. The other pair of chromosomes, and there's only one in human beings, and there's only one as far as I know in all living animals, are the sex chromosomes. Members of a pair of chromosomes that differ between males and females. In humans, you have an X and Y chromosome, or in males, you have an XY. And in human females, you have an XX. Lot of controversy going on about male, female, now trans, and all these other things. Biologically, now we're not going to talk about some of the philosophical things about gender, but biologically, in 99.9999999% of the cases, well, that may have been too many nines. Maybe nine. We'll just say in 99% of the cases, human beings are born with either an X, Y. Excuse me, just a moment. Human beings are either born with an X and a Y, or an X and an X. If you have an X and a Y, you are biologically a male. Now, there are some philosophical things that we can talk about. That can be talked about. And there are some other things that can play a role into gender identification. But biologically, if you have an X and a Y, you are a male. If you have an X and an X, you are a female. Now, that's 99% of the cases. In 1% of cases, human beings have been found to not have this combination. There are combinations where people have been born with 2 Y chromosomes. A lot of misinformation on the internet out there about people, men, who have an extra Y chromosome. For a long time, the extra Y chromosome was called the Superman Superman genotype. And these were people who were either going to grow up to be CEOs of Fortune 500 companies, or they were going to be mass murder serial killers. Yeah, almost all of that has been debunked. There's no value to have an extra Y chromosome. There are also females out there who have an extra X chromosome. That is a condition known as Klinefelter syndrome. And while an extra Y chromosome doesn't give you any benefit at all, an extra X chromosome can cause some real problems. We're not going to go into those details in this course, but if you want to know, just simply type in Klinefelter syndrome. It's Klienfelter, I believe. But if you get close, your search engine will find it for you. And you can look at talking about women who are born with that extra X chromosome. There's also a condition called Turner's syndrome, which are women who are born with an X chromosome. They do not have a second X, but they also don't have a second Y, or they don't have a Y. They're simply born with one X chromosome. And their genotype is listed as X0. And again, not going to go into the details of that. You can look it up if you want to, if you're interested. You can look it up on the internet. Now, I will say this: the extra X or the deletion of an X at the current research shows that there's no relationship with that and sexuality. There are just as many women who are lesbians as there are heterosexual women. So again, just because you have that extra X or the you lack the X, there's the second X, there's no relationship. Karyotyping reveals the characteristics of an individual chromosome. A karyotype is an image, a picture of an individual's complete set of chromosomes arranged by size, length, shape, and centromere location. And I would be almost willing to bet you the next slide is going to be a picture of a karyotype. Yes, that is a human female karyotype. You see, there's chromosome number 1, chromosome number 2. And again, some of these things will look a little bit slightly because these have to be teased out. You have to find a human cell that's about to separate. And then, well, again, with very fine instruments, you have to start manipulating and pulling those chromosomes apart and lining them. They don't line up like this automatically. You have to make them look like this. But if you look here, here's the 2 X chromosomes. So here's the 22 autosomes and the 1 set of sex chromosomes, and you'll see there's nothing there in the Y. There's a chicken female chromosome, and you see chickens have 21 pairs of chromosomes, but chickens don't get their sex chromosomes are not called X and Y. They're called Z and W. I don't know why that was chosen. I'm not a chicken farmer. But you see all the same chromosomes until you get here, and then you see the Z and the W. Don't know why it didn't show us a nice picture of a human male. But in a human male, what you would see is 1 X chromosome and then a little bitty piece of genetic junk, Y chromosomes about this big or so, and it's sitting there. And that I joke with my students all the time. It's that one little bitty piece of genetic garbage that makes a man. That's what makes a man, is one little piece of genetic junk. Men, don't be offended. It's a joke. Then there's the chicken. DNA replication is a process by which a cell copies its DNA before it divides. So again, inside the nucleus of the cell, all of those 46 chromosomes, chromatids have to duplicate. So when a human cell gets ready to duplicate, 46 times 2, there's 92 chromosomes inside that nucleus just begging to get out. Each strand of the DNA helix serves as a template for synthesis of the new complementary strand. It results in 2 double-stranded DNA molecules, identical to the parent. And we have what is called semi-conservative replication. There was a movie made, I want to say back in the 90s. Michael Keaton, I think, was the one that was in it. It was called Multiplicity. Probably available on Amazon Prime or Netflix, or it's probably available somewhere on some streaming service. And it was a kind of a cute movie about a guy who worked really, really hard, wasn't spending enough time with his wife and family, and he decided to have himself cloned. Now, one of the problems with cloning and one of the problems with replication is how much of the original is being duplicated. Now realize, yes, we have to duplicate the whole thing, but how are we doing it? And what happens is the DNA splits, and the 2 new pieces of DNA have old stuff and new stuff. Each one will have an old side and a new side. We are conserving the sides. Now, semi-conservative, because part of the new strand of DNA is completely new information. If you think about a copy machine, you put a piece of paper on a copy machine, you make a copy of it, that is fully conservative replication. You get your original back. A problem with that is if you put a piece of paper, if you put a picture onto, somebody makes a drawing, and you're going to make a copy of it, and you make a copy of it, and then you make a copy of the copy, and then you make a copy of the copy of the copy. By the time you get to 12 or 15 copies, the thing is not really recognizable anymore. Because whenever you make something, whenever you make a copy of something, it's not going to be a perfect copy. there's going to be mistakes. And so the mistakes go from original to copy number 1. Then when you take copy number 1 to make copy number 2, all of those first set of mistakes are carried into copy number 2, plus a whole bunch of new mistakes. Then when you use 2 to make 3, you've got all of the number 1 mistakes, all of the number 2 mistakes, and then mistakes are made in number 3. You play that out 15 or 16 or 20 times, and you get to a point where you've got something that's basically unrecognizable. That's what was happening in that movie Multiplicity. Michael Keaton went and made himself a copy, and the second copy, after a few minutes in the movie or a few scenes, decided, hey, I'm only doing part of, this guy's staying at home with his family all the time, and I'm working, and all I'm doing is working, and then I have to come home and do all this other stuff. So I'm going to go get a copy of myself made. Well, copy number 3, original copy had some issues. Copy number 2 had some additional issues. And by the time copy number 2 went to have copy number 3 made, copy number 3 couldn't even walk and chew bubblegum at the same time. So those mistakes built up. That is fully conservative. This is semi-conservative replication. You keep one strand of each molecule as the parental or the old, the other is new. During DNA replication, the double helix unwind. A set of enzymes called DNA polymerases use the strands as a template to assemble new complementary strands of DNA. Each type of DNA polymerase requires a primer in order to initiate DNA sequences. And then another enzyme called DNA ligases seals any gaps it forms in a continuous strand. And I'm sure there's going to be a picture of this at some point. DNA polymerases, again, are a replication enzyme. They assemble a new strand of DNA based on the sequences of DNA. If there's an A on one side, there's going to be a T on the other side. If there's a T on one side, there'll be an A on the other side. If there's a C on one, there'll be a G, and if there's a G, there's a C. Those complementary base pairs. And again, any breaks in the double-stranded DNA are sealed by DNA ligase. There's my picture. I knew it would be here somewhere. So here is my DNA polymerase. Here's my original DNA. This way. This breaks off, and a DNA polymerase will bind down here and start moving this way, following this. Look, kind of looks like a zipper. As it's undone, one DNA strand is going this way, another DNA strand is going this way. This is called the leading strand. This is called the lagging strand. What this strand has to do is this DNA polymerase has to get up here and get out of the way. Then this one clips on down here and runs and makes a few bases. And then it releases, it comes back up a little farther, and makes a few more, and it releases, it comes back a little bit farther. And then these places where they don't show it here, but here you see this red line is not attached to that red line. These segments, these little segments like this, are called Okazaki fragments. And those are names for short, discontinuous little pieces of DNA that appear during replication. And what will happen is a DNA ligase will come in and actually bind this to this, so you've got a long continuous strand. This strand will continue to move up here as long as this enzyme is unzipping, and this strand will continue to come down here, get to a certain point, it'll release, come back up here, and come back down. We wind up, again, our original is blue, here's our blue side, the new stuff is red, and there's the red side. How mutations? Everybody's heard of mutations. Lots of movies made, especially in the 60s and 70s, about mutations. Proofreading by DNA polymerases corrects most base paired errors during DNA replication. However, occasionally an error is allowed to stay for whatever reason. These uncorrected errors in DNA replication are what are referred to as mutations. And a mutation is a permanent change in the DNA sequence. That change is there when that cell gets ready to replicate. It will pass that mutation on to the next generations of cells. Lots of different things can cause mutations. Different types of ionizing radiation, such as X-rays, most UV lights, and gamma rays. They knock electrons out of atoms, and if you remember, when we knock electrons out, those things are called free radicals. Free radicals normally aren't very good for us, but those can also lead to breakup of the DNA. Most non-ionizing radiation, that's UV light in the 320 to 380 nanometers range, don't worry about that. Just remember non-ionizing radiation. Forms nucleotide dimers that taint DNA and increase the mutation rate. A nucleotide dimer is when a nucleotide forms, and instead of being that 2 in 1 grouping, you have the 2 in 2 grouping, which is going to make the DNA wider, or a 1 in 1 will bind, and then you have this uneven sides to your DNA. Ionizing radiation knocks we talked about that. Chemical groups in tobacco smoke transfer methyl groups, CH3s, to nucleotide bases in DNA, causing mispairs during replication. This is one of the things that happens in folks that smoke and wind up with mouth cancers and lung cancers and all those different kinds of cancers, because the chemicals in the tobacco smoke have altered the DNA, and those DNA mutations go from generation to generation in the cells of that person. Breakdown products of many environmental pollutants will bind irreversibly to DNA, causing replication errors. So again, those environmental pollutants are usually chemicals of some sort. Effects of ionizing radiation, you can see in several different places here. You've got this gap right here where there should be together. You've got the end of this chromosome missing. You've got a gap here. You've got a gap here. All the little red arrows are pointing to areas of the DNA that has been altered by ionizing radiation. 2 advantages and 2 disadvantages of animal cloning. Advantage, if you have the technology, you can continue to reproduce organisms without the benefit of having to have a male and female. If a particular animal, again, Tracker or Liz the cow, wasn't it Liz? I think it was Liz, have very good qualities, you can ensure those qualities go from generation to generation if you are cloning. Disadvantages, with our current technology, there are some health risks associated with cloning, as well as possibly a shortened lifespan or something like that. How can karyotypes be used to advise parents or potential parents about their offspring? Let's say, for instance, you have a condition that you don't know about. It might be that a couple of folks could have some blood karyotyping done, and they would find out that they were carriers for a specific disease. And maybe it's a disease that I'm a carrier for, but I don't show it. My wife's a carrier for it, but she doesn't show it. But we find that out, then we could be counseled on, okay, look, if you decide to have children, there is a 25% possibility that you're going to have a child that has this terrible condition, whatever it is. Some of the muscular dystrophies, for instance, are carried that way. There was a young boy that had muscular dystrophy, was on the Oprah Winfrey Show very, very early, had severe it's Duchenne muscular dystrophy. And he had it, he has since passed away, but he was on the Oprah Winfrey Show several different times. And he got it from his mother. His mother had it, but his mother did not have as a severe case of it as he did. And his mother actually had 3 or 4 children, and all of them died at a very young age. So again, with modern technology, we can look at that. We can counsel people. Yes, you may very well pass this on to an offspring. So it's something to think about before you start to have children. What would karyotypes not show? Karyotypes not going to show if your child's going to grow up to be a mass murderer. Karyotypes not going to show if your child is going to have, be exceedingly tall, some of those types of things. If you have any questions, please email me either at trussell@uaptc.edu, or you can always send me a message through the course contacts, or through the contact link in the course messenger. Have a great weekend. See you next time.